And this is the cochlea.
That is a good model.
And then we're seeing some internal structure, which is going to look like something over there. Here are the three middle ear bones. This attaches to the tympanic membrane. This is not on our test. This one attaches to the oval window. So when the tympanic membrane vibrates, this synovial joint causes this thing to move in and out real fast. And this attaches right there at the oval window. Okay, so this thing is going to push in and out on that oval window, which is going to cause fluid to move inside of these spaces. Okay, now that we got that down, let's take a look at this here. You're going to see that this and this are the same. You see that the cochlea is really a helical structure made up of this device, right? Here's where it starts, and then this goes around and around and around. They cut it away, and that's what we see in the middle. You agree?
Mm-hmm.
Okay. This is called the organ of Corti, or the spiral organ. It consists of three fluid-filled spaces. This fluid-filled space is in contact with the oval window, and the stapes, the last bone, pushes against it. There's the stapes. Okay? That little bone there. When fluid then is pushed into here by the movement of the stapes, it creates a pressure difference here that causes this membrane to vibrate. And I'll tell you all the parts in a second. When this membrane vibrates, these cells, these in particular, have their tips rocking in this gelatinous roof. And when the tips of this cell rock in the gelatinous roof, it opens ion channels and depolarizes the cell. And then the cell will send off action potentials. You see a ganglia here. This is fluid filled, and when fluid moves in here, it deforms this membrane. Pressure of fluid moves right through this thin membrane and deforms this one. Eventually, the fluid movement in here will come around to this side, and this side then will dampen off at the round window. If none of this makes sense, hang on and we'll be looking at it in lecture. This is called the scala vestibuli, and these two both get the name scala. Scala is a passageway. Scala vestibuli, it is in contact with the vestibular area of the inner ear. There is the oval window, so this big central opening in here is called the scala vestibuli. Okay? It's filled with a fluid. That same fluid is in here. This is called the scala tympani. Okay? This one could be called the scala media, but on this model, it is called the vestibular duct. Pardon me, coccular duct. Look, find the coccular duct. Okay? All right. This very, very thin membrane that separates the scala vestibuli from the cochlear duct is called, I got a cheat, is called the vestibular membrane. Scala vestibuli, vestibular membrane, cochlear duct, scala tympani. This is the basilar membrane. This is a bone, and then the basilar membrane completes the closure of this passageway. On the basilar membrane, there are a number of cells. Some of them are supporting. Some of them are referred to as hair cells. The hair cells are shown here with these 9s and these 8. The hair cells have something called stereocilia on their surface. The stereocilia stick up into the fluid that is found in the cochlear duct. And the tips of the tallest stereocilia stick into this membrane up here called the tectorial membrane. Vestibular membrane, basilar membrane, tectorial membrane. The tallest of the stereocilia stick in the tectorial membrane. When the basilar membrane vibrates, this causes the stereocilia to rock in place. And this is going to lead to changes in potential across the membrane. The central cells are what are called hair cells, and then the cells that surround are called supporting cells. The outer 3 hair cells, thank you, I had it and I left it somewhere, I don't know what I did with it. The outer 3 hair cells are here, the innermost grouping of hair cells is here. These follow the spiral organ all the way through the cochlea. Just between you and I, some portions then of the spiral organ detect low frequency noise and some detect high frequency noise. Any noise that comes into the inner ear that does not deform this membrane is not heard by a human being. So this is ultra high frequency noise and ultra low frequency noise we don't hear because it does not alter this basilar membrane. It comes in, it creates pressure, but it does not affect the basilar membrane. Okay? I don't know there's too much else to tell you.
Bony labyrinth.
The bony labyrinth is on this model over here. If and when you study the inner ear very carefully, you're going to find that the inner ear exists in a space in the temporal bone. The name given this space in the temporal bone is the bony labyrinth, a series of rooms inside of the bone itself. The word is, you couldn't cut into that temporal bone and you remove this because it's, remember what's inside a bone. There's going to be lining a bone. There's always going to be, or, you know, on any surface of bone, there's going to be periosteum. You know, there's going to be endosteum inside a bone. So all I'm trying to tell you is that the bony labyrinth means that there's a space in the bone where this thing lives. Okay? That's really long-winded description, but it can be a little confusing.